So TLRs, which stands for toll-like receptors, are a type of receptor that span the membrane of a cell, usually certain types of white blood cells. Pathogens or disease-causing bacteria usually will have antigens, which are like specific structures on their outer surface. TLRs are able to recognize these structures and identify them as non-self molecules, meaning it's a bad bacteria that we don't want in our bodies. After they recognize these non-self molecules, TLRs will send out a signal to our immune system, telling it where to send certain cells to fight the bacteria. So TLR4 specifically recognizes a structure called lipopolysaccharide, or LPS, which is found on the outside of certain unwanted bacteria. TLR4 can't recognize LPS on its own, though. There's a protein designated to bind to individual parts of LPS and take them to another protein called CD14, which then takes it to TLR4. Another protein called MD2 binds to this complex to help TLR4 recognize the LPS. Once MD2, TLR4, and CD14 are bound to LPS, another TLR4 comes to join it, and there's a change in the conformation, meaning the shape, of TIR domains inside the cell. This signals another protein, MyD88, to bind to the receptor, which then leads to a really long and complex signaling cascade, which eventually warns the cell of the pathogen. One main event in that cascade includes nuclear factor kappa B, or NFKB, which is a complex of proteins that controls production of signaling cells and making new DNA. NFKB has inhibitor kappa B bound to it, which stops it from functioning. Once LPS is bound to the TLR4 complex I previously explained, the inhibitor is released and destroyed, so NFKB can become active and eventually lead to immune responses, such as inflammation, so that the pathogen's growth can be blocked.